Well, this project is about analyzing sugars, and I thought I'd remind everybody how sugars actually get into plants, and that's through the process that actually takes up CO2 out of the atmosphere. So we have usually carbon dioxide and water that is then turned with the help of sunlight into glucose and oxygen. And then the glucose is the sort of principal chemical that's been sort of the first product produced out of photosynthesis, is then converted into other sugars such as fructose here or sucrose, which is the main transport sugar that we have in plant cells. But we also have stored sugars, which is starches, for example, that will then usually be stored in plant cells, in plant leaves, in the chloroplasts. So together, these sugars and starches, soluble sugars and starches, are called nonstructural carbohydrates. And if you then look at what happens with nonstructural carbohydrates in plants in general, you'll see that they're central to almost anything that a plant will ever do. Because they are a form of chemical energy that the plant can then use later on, be it for metabolism here, be it for transport, getting that sugar that's been produced initially in the leaves to the areas where it is needed, either, for example, in a fruit or in a root system. It's also used for osmoregulation. That means defense systems against drought stress, where plants accumulate solutes in their leaves to be more drought tolerant, or it's exported, in this case, for example, into fruits, or they're used for certain biosynthetic products. On the other hand, non-structural carbohydrates can also be moved into storage organs and therefore be available for later use in plants. And one thing that we don't really know, and you can see that from the different colors in the arrows in this graph, is whether some of these processes are active or whether some of these processes are passive. So, for example, is starch or non-structural carbohydrate accumulation happening because it is an active process that the plant is actively increasing the amount of starch in it, or is it simply an overflow process where non-structural carbohydrates are accumulating in the plant tissue simply because they're not used anywhere else? So sugar doesn't have to be exported, or the plant is not growing, or fruits are not growing, and therefore it goes into storage. So as we can see is that non-structural carbohydrates are very important energy stores for plants, and the dynamics of non-structural carbohydrates can have quite significant impact of plant growth, crop load, fruit yield, and fruit quality as well. So in other words, we need to get a good understanding about the dynamics of non-structural carbohydrates in plants, and in particularly in horticultural crops. Non-structural carbohydrates are also important in sort of more ecological questions around tree drought mortality, where they're thought of as a fairly important energy store that plants can run out of and effectively die of carbon starvation. So it's critical to have a good, accurate, fast, and affordable measurement for non-structural carbohydrates. So this is where this project comes in. If you look at some results published last year in the journal Tree Physiology, and just look at the top two graphs that we have here, it's the concentration of starch or total non-structural carbohydrates of Prunus persica leaves. So Prunus leaf, peach leaves, that were analyzed by 30 different laboratories worldwide. Okay, I let that sit for a moment. It's the same material that was sent, to 30 labs. And of course, they use slightly different methods. So these labs here used acid digestion of the starch. These labs here used amyloglucosidase to digest the starch. And these labs here just used alpha amylase and amyloglucosidase. But the level of output that we're getting was quite phenomenally different. And the same is true for the non-structural carbohydrates, whether they're being extracted with water, or with ethanol in water, or with methanol chloroform in water, or with ethanol alone. The answer was that it's not the technique that was the big difference, it was just you could not compare the results from these different labs. It's quite astonishing. And I mean, it was published, I was aware of the study, we didn't take part in it, but it is quite remarkable that you have the same material analyzed by 30 different labs in the world, and one of them you measure, oh yeah mate, that starch concentration is about 5 milligrams per gram, and the other lab comes out and says, no, it's 120 milligrams per gram. so a factor of 120 more. So it's really quite astonishing. So it calls for faster and probably more accurate measures of non-structural carbohydrate analysis, because a lot of people around the world now have lost faith in, well, can we actually believe what figures we're getting if somebody comes up, oh, starch concentration is 2%. Well, it may be 2%, or it could as well be 10%, depending on what lab you send it to. The other issue is, of course, that the chemical analysis, although you can identify, should be able to identify with a high degree of precision if you are a lab with good standards, so you can repeat analysis, is that they're quite costly and they take a long time. So it's not like that you have a very fast turnaround time. So one technique that we now want to investigate in this project is mid-infrared spectroscopy, which has been around for a fair while in sort of food chemical analysis. It's been used in the last few years also for soils analysis, where you do scans with MIRS to look at soil carbon, for example. It's a mid-infrared wavelength and spectra from 2 to 25 micrometers, where the specific molecular vibrations are sensed and then associated with the functional groups that are common in the tissue. And I picked an example of a recently published study where people did MIR spectra in frozen grape berries at 5 different developmental stages. And here you have the entire spectrum of an MIR spectrum. So here's the big water peaks that you have here. And then this is the region of interest where people want to look a bit closer. The grapes were either green, pre-veraison, at veraison, or then ripe or in post-veraison classes. And then they compared these results. So you can see from the different absorption patterns here in this region also that something is changing in these grapes. And in this case, this is the area where glucose and fructose are absorbing. So you can see that as the fruit is ripening, the concentrations of glucose and fructose are increasing. And when they did analysis of partially squares regression models, it's a complicated way of analyzing these spectra, then and compare that to chemically analyzed data of glucose and fructose. We have this data here in the right-hand corner. You see the reference material analysis and the predicted fructose and glucose analysis, and the correlation is actually very, very strong. So it gives good hope that we can actually use the MIRS to analyze sugars in plant material. So the analysis of an MIRS, it's a reasonably expensive instrument for people who want to buy it. You have to fork out probably more than for your average car. But the benefit is that all you need to do is grind your plant material to reasonably fine powder, put it in your MIRS, do a 2-minute scan, and you're done. Then the major process is then in analyzing the data and actually doing this kind of partially squares regression in order to come up with the regressions to see whether it's working. So what we will do is we will calibrate the MIRS instrument that we have either at Degeter at MacLeod or at the University of Melbourne in Creswick with material that we have already analyzed for sugars from an experiment that we run with different eucalypt species, actually here at the Dookie campus of the University of Melbourne. We've grown 20 different eucalypt species, we've harvested them, took root samples, stem samples, leaf samples, and branch samples, and they're already analyzed for their sugar concentrations and starch concentrations. And then we're validating the MRS method in an horticultural context with tissues from an experiment around the back here with stone fruits that have been undergoing different crop load experiments, so they've been having different amounts of crops in there, and Dario Stefanelli has been taking samples at different times of the year to analyze whether the crop load and the success of the fruit production is actually correlated to the concentration of sugars in the samples. So the outcome of this method will be a really robust evaluation of an innovative method to see, can this MRS spectroscopy really enhance our ability to analyze sugars in plant tissues? What are the pitfalls around it? What are the difficulties of doing this with different materials? Does it work in peach? Yes, no, it doesn't work in eucalypts because something else is absorbing at the area where we have glucose or starch absorbing, and then that is a major limitation. So the application would be probably not so much for the average grower because of the instrument being still quite expensive, but probably more in a research context to find out what are ideal crop loads, what are our ideal irrigation schedules. The project that we heard this morning around irrigation, of course, this will all have an influence on the amount of sugars, how much sugars are transferred actually into the fruit, and then as Virginia told us about the quality of the fruit And sugars are key to this. So I personally probably see this project more as a research application, but if it would work, I think it will revolutionize the way we can do sugar analysis in an horticultural context, because we can do it very cost effective, because the actual analysis doesn't cost really anything other than you picking a leaf, grinding it, putting it in MIRS, and then doing the analysis of the spectra to the concentrations.